Dear all, here we are with Annamaria Mocciaro, who comes from Italy and got her degree in biological sciences from the University of Palermo. And for the PhD, she moved to Germany to the laboratory of Professor Elmar Schiebel, where she studied the cell cycle regulation. And she joined the laboratory of Professor Michael Rappe at the UC Berkeley on the identification of ubiquitination enzymes involved in the regulation of stem cell differentiation. And she is also a fellow of the CIBER Stem Cell Institute and the American Cancer Foundation. Thank you, Annamaria, for being here and for accepting our invitation.
Thank you for asking me to do this interview.
So, as a first question, could you please explain the state of the art and the research avenues in your field, taking into account a landscape up to 2050? So, if you can explain which kind of stem cells exist and what they are used for.
Of course. So, there are mainly two different types of stem cells. Let's start saying that the main characteristics of stem cells are self-renewal and differentiation. Self-renewal means that stem cells can actually duplicate themselves exactly, and then differentiation means that they can differentiate in many different cell types. There are two main types of stem cells. One that are called adult or tissue stem cells, and that are the ones that are present in organs, are responsible for the renewal of organs during lifespan of an individual. And those cells can differentiate only in a certain type of cells. Like, for example, cells that are stem cells of the blood will produce blood cells, or for example, stem cells like the mesenchymal stem cells will produce bone, cartilage. So these cells are less potent, we would say. So they can differentiate in not so many types. Other cells. The second type of stem cells is called pluripotent stem cells, and those are the ones that can differentiate virtually in any type of cells. Pluripotent stem cells can also be subdivided in two more classes: embryonic stem cells and induced stem cells. So the embryonic stem cells are the stem cells that are taken from an embryo and that basically can be propagated in vitro. The induced pluripotent stem cells are maybe at the moment the most famous because they were generated through a discovery that then granted the Shinya Yamanaka the Nobel Prize for Medicine in 2012. And those cells come from a reprogramming event, meaning that you take cells that are already differentiated, like fibroblasts, for example, and then you infect these cells with 4 different factors called the Yamanaka factors that can basically bring them back to a stem cell condition. So you go from differentiated cells to undifferentiated cells. So that's why you reprogram them back. So you don't need an embryo to actually produce these cells. This, of course, comes in handy because in theory you could, you won't have all these ethical issues that are associated with human embryonic stem cells. So these are the main types. And at the moment, of course, in the field, there is a lot of research going on at the basic level, but also at the applied level. Most of the research for induced pluripotent stem cells is focused on trying to characterize them better and to see if they are really as similar to embryonic stem cells as they look like. So from the genomic point of view, from the gene expression point of view, morphological point of view, they seem exactly like embryonic stem cells. So they look very promising because that might actually be a way, inducing pluripotent stem cells might be a way to obtain large amounts of stem cells without having to isolate them or derive them from an embryo. And another very interesting thing of induced pluripotent stem cells is that you could derive them from cells of different patients, which means that you could use them for therapy and you wouldn't have all the issues of immunosuppression that, of course, you have when you transplant heterologous cells into an individual. So they are very, very fascinating, and it looks like the field is moving more from mouse embryonic stem cells or human embryonic stem cells to induced pluripotent stem cells, but still there is a lot to be done in that department at the level of like basic research and characterization of these cells. So, of course, I mean, if we look at the research in the past like 10, 15 years, even longer, maybe 20 years, and then if we look at the future, we can easily say that while tissue stem cells, so adult stem cells that are the ones that can differentiate only in one type of cells, has been used therapeutically for many years already, since the 90s probably, especially in the context of hematopoietic stem cell transplantation or stem cell-based skin grafting, where you actually insert stem cells to repair damaged skin because of burns or other offense. So this has been going on for quite a while, but the use of pluripotent stem cells to develop like personalized cell-based therapies is a relatively new approach, and it's also a partially still unexplored field. On one hand, stem cell research has the potential at the moment to improve drug discovery and accelerate preclinical toxicological assessment of new drugs, because when you induce differentiation of human embryonic stem cells and then you expand differentiated cells after induction, in theory, you could have unlimited supplies of defined human cell types that could be produced in the lab and could be used for testing of new drugs, for example. So this basically would allow the primary drug screens that at the moment are conducted on, for example, animal cells or human cells, but they are mostly transformed cells, for example. I mean, the screens could actually be not only tested on human cells, which would reduce in the end the risk of promising drugs that fail the final phase of clinical trials because there are differences, for example, between animal cells and human cells. This would also reduce the amount of animal testing that you need to actually be sure that a drug can be then used on patients. So, moreover, there's also to say that when you're actually using induced pluripotent stem cells that, as I said, can be produced from cell lines coming from different patients, for example, you could carry on different parallel high-throughput screens for drugs, and you would compare different cell types coming from different patients, and then the screening process would like minimize the variability in the response of the same drugs that are coming from genetic differences from different patients. So it would, in the end, speed up the process of reaching a working drug that can then be put on the market or tested directly in the late phase of clinical trials. So basically, at the moment, probably in the next 10 years, the let's say closest application of stem cell therapies will probably be in drug development for developing like personalized cell-based therapies. Another very, very interesting approach that can be used with induced pluripotent stem cells is to reprogram cells coming from patients, for example, that have autoimmune disease, hereditary disease, and if you reprogram cells coming from, for example, diseased patients to iPSCs or induced pluripotent stem cells, and in comparison, you also maybe use the cells of a relative that is not affected by the disease, and then you also de-differentiate these cells, reprogram these cells into induced pluripotent stem cells. You could compare, for example, the behavior of cells that will develop a disease and cells that won't develop a disease, although they're coming from people who are related. And then, of course, you can obtain new information about how the disease develops and what is different at the pluripotent stage between cells that have disease and cells that don't.